Thank you so much. Well, thank you so much for having me. It has been a blast to be in this meeting, so interesting. So I am interested in understanding what aging has to do with Alzheimer's disease, another neurodegenerative disease, and we have talked quite a lot about changes around aging. And if we define as the World Health Organization what aging is, they will say something like there is the accumulation of molecular and cellular damage that will increase the probability of suffering disease and eventually death. Not very appealing, but to make sure that seems to be quite right. As we age, we have a very long, flat timing in terms of chance of death until we are like 60 or 65, and then over 70 to 75. That's increased exponentially in our chance to die. And if we go in terms of functional decrease, that function decline is much earlier. And we are used to think that aging occurs after we are 60, and it does not. It starts probably in our 20s. And one of the first things that age in us is our eyes. So aging in our cells occurs along our life until we notice probably when we have a decline that is strong enough as to make things harder. In the nice part, our brain age is much less later, in part because we are able to acquire experience, and then our decision-making and our ability to approach complex problems, in fact, increase as we age. If we think about these different diseases, arthritis, heart disease, cancer, diabetes, and if you think that everything goes down the drain, you can be happy because asthma goes down with aging. So there you go. You have asthma, you are on the right track. But for dementia, it's more or less the same. These are numbers from Chile, and you can see that very similar to what you will find in any European country. We have an increase in the appearance of dementia. 65% of them will be Alzheimer's disease. That goes up, being more prevalent in women than in men, as we haven't noticed before. So what's the important part of dementia? There are many sides of that. One is the cost and the impact it has in the person's life, in the family life, and society. It's very costly. For every patient, we will have a Rita, we will have someone that will take care of this patient for a couple of decades. In terms of the mechanism, so far the most popular view of Alzheimer's disease has been the accumulation of A-beta. A big deal of the research has been done in that sense, and to make sure most of the therapeutical tools that we have come out is surrounding the area of decreasing A-beta. Needless to say, those therapies have not been successful. Lately, we have 2 antibodies that have been approved by the FDA in the United States, but to be honest, they have been proved to be able to remove A-beta very nicely, but so far there is not clear evidence that there is any change in the progression, in the amount of deterioration, and even in the age of death of the patients having or not having this treatment. So for our view, we decided to turn a couple of decades ago into trying to understand what's going on with aging, since aging is the main risk factor, at least for sporadic AD, that accounts for 97% of the patients. And what has been interesting is in the last decade or so, it became very clear that in terms of epidemiology, the risk factors that you will think about cerebrovascular or cardiovascular disease are more or less the same that you will find in some of the dementia mechanisms. And that's interesting because it's broadening the view that probably there are more general things that will be relevant for the appearance of the disease. So we'll have many aging-associated changes, increased inflammation, increased oxidative stress, and change in the immune defense, decreased adaptive and increased innate immune response, in fact. And also several of the risk factors and conditions promoting inflammation, as it is high blood pressure, insulin resistance or diabetes, dyslipidemia, hypercholesterolemia, all those are also risk factors for Alzheimer's disease. And inflammation is a key word because, in fact, aging itself, it's inflammatory. We become inflamed as we age, and all these chronic disease, in fact, they don't do anything but increase as well the levels of inflammation. So aging under it's a major risk factor, and for us, it facilitates the development of the mechanisms that will lead to dementia. They are not the same, but it's a facilitating mechanism. And there is another element it's very interesting or very important for our research is the fact that has been mentioned as well, that we have a disease that occurs in decades, 2 or 3 decades. That means that whatever is meaningful for the disease to develop will not occur at 80 or 70, not even at 60 years old, but probably will be occurring when you are 40, 50, 55. So in that sense, that's a fact that's important for our research, because maybe whatever is initiating the disease occurs decades before, and many other changes will occur as to compensate or as secondary damage. So what we are required in terms of understanding the disease, and definitely in terms of defining a good therapy, is to be able to target this very early event and not be fooled by many other things that happens along the way. There are 2 papers. There's one paper is very nice. Many of you were not even born by that time. We are talking about 1998. And this is a colony of rhesus monkeys that were 28 years old. So what they did is to inject a very small amount of fibrillar A-beta. What they observed in these monkeys is that they developed plaques that were very similar to Alzheimer's disease patients. They will have aggregations of microglial cells. They will have dystrophic neurites, and they also will have hyperphosphorylated tau. If we see the numbers show that all monkeys, not young ones, are the ones that have bigger plaques, more phosphorylated tau, and more activation of microglial cells. And if we compare among species, rhesus is a primate, an old world primate. Marmoset is a new world primate. Both are primates. And you see that rhesus monkeys has a much higher response. That means that it's a species-specific response. And what's very interesting for us is that rats have a very low response, and most of our research is being done in rats or mice. So for our work, we propose a changing paradigm for Alzheimer's disease. So we view A-beta as a consequence, and we think in our hand that the regulation of microglial cells, of activation of microglial cells, could be relevant for the mechanism, the pathogeny of the Alzheimer's disease. And we, in fact, think that TGF-beta could be relevant for that dysregulation, and I will tell you why. So depending on this regulation, you will have increased accumulation of radical species and inflammation. Of course, directly you will have change on the neuronal function. And both oxidative stress and inflammation is pro-amyloidogenic. So it increases the production of A-beta and the aggregation of A-beta. And of course, A-beta inflammation or radical species will act on neural function. We could start having that synaptic dysfunction in terms of a more functional, reversible effect, and eventually it will lead to neurodegeneration. So we are not denying that A-beta plays a role. We just think that this role is played down the river. So if I attack this A beta, there are many other things that are still happening, and the patient will be able to progress without change. Of course, all this interpretation, when you have this very long-lasting chronic disease, are open, and many things could explain the different results. So we propose that, not very original, that there is less protective and more cytotoxic activation of microglial cells. And we think that aging is promoting this effect because you have inflammation, hypoxia, metabolic dysregulation, and injury. There are many things that are going on in terms of the activation of these cells and potentially modifying their activity. Very simple results in terms of the activation or the way these cells talk to each other. We think that always a crosstalk between microglia cells, in this case with astrocytes, and we see that in general, when we have mixed glial cells compared with microglial cells, the activation by inflammatory molecules, LPS, interferon gamma, APP, beats, A beta, are the combination of these different factors. You have sometimes no difference, but often a certain effect. When you do this combination with neurons, in this case hippocampal cells, we can see that at least for some activation inducers like APP and A beta 142, you almost have no response to them. It's very similar to control. But when you use LPS interferon gamma, very strong inflammatory reaction, the presence of microglia cells induce the production. In this case, we're evaluating nitrites as a stable metabolism of nitric oxide. So I will just show you like major results we have. The differences in the morphology of the cells have been long described. Cells from the hippocampal of 18 months old mice look really with big cell bodies, coarse processes, and even if Marina doesn't like it, that's the typical morphology it's described for activated microglial cells, and it's very similar to the morphology we'll see if you use inflammatory mediators. But there are also functional changes. There is a decrease in the regulation of inflammation. There is a decrease in the activity of TGF-beta. That's what I will show you next. There is an increased production of radical species. So confronted to the same inflammatory activation, the cells, depending on the age, will respond differently. And what's very interesting, they do have phagocytic activity, no difference. But if you try to induce additional activity with LPS or TGF-beta or beats, you will see that these cells in general are not able to upscale their activation as a young microglia cell will do. And what's even maybe perhaps more interesting is that even if the phagocyte, the degradation machinery and at the level of lysosome is downregulated, that means that cells may be phagocyting, but probably they will be less efficient in the ability to get rid of whatever they're eating. And usually they don't speed it up, but they remain activated. And when they remain activated, they just recruit more and more microglial cells. So they have a very important reaction of massive activation of cells. So what I will show you briefly, why we think that TGF-beta signaling is important and how it changes as we age, at least as mice age. I don't know if we age. Scavenger receptor A, that also change with aging, what's the impact it has, and just a bit what we're doing now, neuromicroglia crosstalk, and that will be fractal kind pathways that I think is very interesting in terms of the dialogue or the regulation of microglia via neurons. So all the experimental approach we use in the lab, we try to use in vivo or ex vivo samples. In vitro, ex vivo, we do neurobehavioral testing, serology to count plaques and cells and whatever is needed. And also we do different techniques in order to evaluate cytokines or different molecules, signaling pathway, etc. And of course, usually we start with in vitro experiments to try to set everything before we go into animals. We developed by congenicity a triple transgenic animal that was a PPPS1 model of Alzheimer's disease, or at least a beta accumulation that was knockout for scavenger receptor 1, and I will tell you why we did that. But starting with TGF-beta, if you see unstimulated cells, there are mice hippocampal samples, you will see that if we compare 2 and 12 months old animals, you'll have a higher level of TNF-alpha as well as a higher level of TGF-beta. So there is an increase in cytokines depending just on age. These are unstimulated animals. If we go and we stimulate these mice with intraperitoneal LPS as an important sign of inflammation, we'll see in blood plasma TNF-alpha increases. That doesn't change with age, it's more or less the same. There is an increase in TGF-beta, plasmatic TGF-beta, which it does goes down as the animal age. And in terms of what the levels we can find in the brain, we will see that LPS does induce a very modest increase in young animals, but it doesn't undergo that increase in the older animals. And finally, in terms of the signaling, we evaluate again in response of LPS what happens with the activation of one of the major pathways that I will just show you in the next slide. That's the SMAD pathway, and we are measuring SMAD3. So if at 2 months old, we will see that both the total amount of SMAD3 and the phosphorylated SMAD3, that is the active that signaled activation of the pathway, will be increased in response to inflammation. However, when we have 12 months old animals, we do have, let's say, a basal level, important basal level of SMAD3. It's not increased by inflammation, and we don't have the activation of the SMAD pathway. This is just to make sure that LPS is producing the inflammation we expect it to do. So why this is relevant for us? And that's a general cartoon of the activation by TGF-beta. TGF-beta that dimerized forms an heterotetrameric receptor, which will be able to activate a canonical pathway that's mediated by these SMAD mediators that will go on target genes and increase transcription, and we will have the increase in SMAD7 that will be endogenous stop for the inhibitor for the pathway, and many other things happen, of course. And we choose another one that is a MAP kinase phosphatase 1 that is responsible for the phosphorylation of molecules that are phosphorylated by kinases. So this will be the canonical pathway. This will be mostly related to everything that is regulation of inflammation as well as survival. But what for us was striking to learn, we didn't do it, but to learn that TGF-beta also has this non-canonical pathway. And this non-canonical pathway, it's mediated by the activation of MAP kinase and PI3 kinase. And if you're involved in inflammation, it's clear that these pathways are in fact activated during inflammation. So you wonder why a regulatory cytokine actually it's acting on these two pathways. But the interesting thing is that part of the activation of the canonical pathway will include the activation of this phosphatase, and that phosphatase will be able to dephosphorylate the activated MAP kinases. So somehow in younger age, you may think that this canonical pathway will prevail in terms of the functional effect. But maybe if in aging, when we have increased receptor type 2, we have increase of TGF-beta, but we have decrease in the activation of the canonical pathway, and we also have inhibition of the transcription of MAP kinase phosphatase. It will lose this part. In that sense, in aging, it could prevail this pro-inflammatory pathway, and in that sense, TGF-beta will be unable to serve its purpose of regulate inflammatory activation. We become interested as well, simultaneously, more or less, in these receptors. We were interested in general in receptors, in scavenger receptors. Many of them are known to bind A-beta and have been revealed as relevant for A-beta phagocytosis. This is a plaque for the patient, and you can see that many of the microglial cells that are surrounding these plaques are expressing scavenger receptor A. So we wonder, we were studying different scavenger receptors, we wonder if you have this redundancy, what's the meaning of having all these different scavenger receptors that could be relevant for uptake the A-beta or many other things, because they are pattern recognition receptors. And we thought that maybe they don't only participate in the uptake and degradation, but they could maybe also signaling in terms of what inflammatory response these cells will have. So we began to follow these receptors in that line. Can we have different responses in terms of inflammation depending on the type of scavenger receptor that the cell expresses? In fact, they express many, many different receptors, and these receptors change in their expression with aging and with many other facts. This is a scavenger receptor that binds many different things. It was initially described for the ability to phagocyte microbes, but also participates in the lipoprotein uptake and also different microbial, virus, and apoptotic cells. So it has a very broad function. What we found was that if we compare through the protein content of scavenger receptor A, depending in age, the amount of scavenger receptor A in 12-month-old animals was much less than in younger animals. But what surprised us that using the APPPS1 model for Alzheimer's disease, we noticed that even when these animals were young, they already have lower levels of scavenger receptor A, which is being shown here. And I have to stress the point, why 12 months old? And it's a bit for what I say. I mean, whatever is happening leading to Alzheimer's disease is happening much younger. And mice of 12 to 17 months old, more or less, it will be equivalent to people in their 50s. So that's why we decide to use mostly that age earlier and not in already aged animals. So this was a tissue, so we decided to go for flow cytometry now to see if specific cells, in this case microglial cells or astrocytes, will have more or less the same behavior that we check on the mixture of cells that is tissue. And again, we are comparing wild type and APPPS1 animals, and in the top we have IVA1 cells, microglial cells, and GFAP cells, positive cells for astrocytes. And what see that is more or less the same, what we describe. In the tissue, wild type animals decrease with aging. APPPS1 have already young animals, 3 months old, they already have low levels of scavenger receptor A, and the same happens with astrocytes. And astrocytes for us are very important. We know that they phagocyte many things. They are not professional phagocytes like microglial cells, but there are so many that in fact their impact, they have a good impact in terms of the function. And what surprised us is that now if we measure by quantitative RT-PCR the relative expression of scavenger receptor A messenger RNA, if we compare controls with A-beta, A-beta it's down-regulator for the expression of scavenger receptor A. So you may think in case of the Alzheimer's disease patient, or in this case, this APPPS1 animal, that as soon as they began to accumulate a beta, in fact, they could be further decreasing the amount of scavenger receptor A available. Joining this story with our TGF-beta interest, we found that there is 17 motifs for binding for SMAD3 in the gene for scavenger receptor A, that is MSR1, and we choose these 3 closest, and we did ChIP-seq for SMAD2 or 3, and we cloned this promoter from scavenger receptor A in a luciferase vector, and we proved that, in fact, the binding of TGF-beta, the binding of SMAD3 to these promoters were able to move to change the expression of scavenger receptor A. And what's the effect they will have? That is, again, quantitative RT-PCR, and in red we have TGF-beta, and we see that the presence of TGF-beta decreases the relative expression of scavenger receptor A, but if we add TGF-beta plus C3, that is a specific inhibitor for SMAD3, we see that this change and returns more or less to normal, and similar results we obtain at the protein level. TGF-beta here will decrease the presence of scavenger receptor A protein compared with the pH as control, and again the presence of an inhibitor of SMAD3 will abolish importantly the decrease. So what will be the impact of the absence? Because we decide to work then with these scavenger receptor knockouts as a way to prove what happens when, in fact, this receptor is absent. So we did this in vitro, in vivo. I will show you, yes, our results in hippocampus. We have also in culture cells and in plasma. But in terms of the hippocampus, what we observe, again, we have 3-month-old animals, and we see that there's low levels of interleukin-1 beta and TNF-alpha. There are no significant differences in terms of what happens, or I should say, wild-type APPS1, and here is the knockout for a scavenger receptor, or the triple transgenic animal. That will be exactly the same order for all the graphs. And interleukin-1 beta, TNF-alpha, no big changes, but in the case of interleukin-10, we'll see that animals that were knockouts for scavenger receptor A have much higher levels of interleukin-10, no difference whatsoever in TGF-beta. If we go and compare this with what happens with 12-month-old animals, we see that it's a huge increase in the amount of interleukin-1 beta, not so high in the case of TNF-alpha. In fact, it was a bit lower than what we expected. And what was striking was what happened with the regulatory cytokines. So for interleukin-1 beta, you have this huge, interleukin-10, you have this huge decrease in the amount of interleukin-10 for animals that are knockouts for scavenger receptor A, and that was even more striking for in the case of TGF-beta, that 12-month-old animals knockout, or even the APPS1, have levels that were much lower than what we observe in younger animals.
Excuse me, Rommy, would you be able to start wrapping up? I think we're beginning to run out of time.
Okay, yeah. Give me 2 minutes.
Absolutely.
Since I speak slower. Okay, so very, very briefly, there are clear differences between signal transduction in the animals that are wild type and scavenger receptor A, scavenger receptor knockouts, both young kinase, PIT38, and ERK, And also we found similar effects in the case of NF-kappa beta. So there is a later and decreased activation of NF-kappa beta in these animals that are knocked out. What will be the impact in the whole animal? That's shown here. We are comparing here APPS1, triple transgenic animals. They have much the higher levels of plaques. It was interesting that both the size and the number of plaques were higher in the triple transgenic animals. It was interesting there was no difference on the interaction of microglial cells with the plaques. In fact, if we also measure CD68 positive cells, and they were even slightly higher in APPS1 knockout for scavenger receptor A. So it was no a matter of the activity of microglial cells. It was a matter that removal of management of the plaques was different. And the last thing I was interested showing you is that we also did some experiment much early on showing that in terms of, this is apoptosis, tunnel experiments, and we see that the presence of microglial cells in the hippocampal cultures increase greatly the activation produced in inflammatory conditions. That was observed later. It was not observed very early at 1 day, but it was observed at 4 days. So our interest for fractalkine, it's born because of this, and we were especially interested because it's an off signal for the activation of microglial cells. And here I only want to show you that fractalkine is a cytokine, it's a chemokine that is bound to a membrane. But in inflammatory conditions, it slices and you will have soluble fractalkine. So we become interested what happens with fractalkine with aging, and what we found is that there is a specific increase of the receptor at 12 months old animals. There is an increase of soluble fraction of fractalkine of 70 kD, and also specifically appears a form of 40 kDs that is actually produced by the secretion of cathepsin S by microglia cells specifically. That's metalloproteases, but this is an effect of cathepsin S. This has to do with signaling, and here it's important that we have signaling mediated by NF-kappa beta, but also by NRF2, that is antioxidant, and we think that's very relevant in terms of what will be the final effect. And just our working model, we have inflammation or different types of injury will increase the level of TGF-beta. TGF-beta will act on SMAD3 and MAP kinase pathways. This will, oops, what's going on? Okay, this will have to do with neurotoxicity and cytotoxicity mediated by these cells. MC, oops. Meanwhile, in young animals, membrane-bound fractalkine will be acting on this receptor. In aging, you will have chronic inflammation. You will shut down the activation of SMAD. That will decrease the activity of scavenger receptor, favoring the accumulation of A-beta, but also you will have the production of soluble forms of fractalkine that will have a different effect on terms of the inflammatory regulation of the cells. So thank you very much, and my apologies for being so slow.
It's all good. Would you like to show us your final slide? You had some beautiful pictures.
Well, yeah, that's to recognize everyone that worked here and how everything got made. So
fantastic. Thank you very much. Any questions, please? We have time probably for a couple. Yes.
MKP1 is a member of a family of MAP kinase phosphatases with differential expression, differential recognition of MAP kinase family members. Is MKP1 the only one that you see regulated by aging?
It's, to be honest, the only one we look for, because we were interested basically in TGF-beta pathway. Phosphatase in general, there are very few, they are very broad in their activity, so in general they activate big families of kinases.
And so do you think is the inactivation of MKP1 due to redox regulation, with the increased ROS levels, or is it's an intermediate early gene, so is there regulation of its expression level?
I missed the first part. Which one, the regulation of?
MKP1.
Oh, the same one. Well, I'm sure these phosphatases are activated by different means. That's sure. Probably is not specific for this pathway. What we think is interesting is the fact that when TGF-beta is a very complex way in the sense that everything has to fit together at the target cells in order to be able to signal. So we think that the fact that this pathway is, a SMAD pathway is able to activate phosphatase, it means that in temporal means will be occurring at the same time that you're activating the MAP kinase pathway. So probably the relevance will be in that sense. That's why I think it's very important, the activation of that phosphatase for SMAD pathway.
Brilliant. I'm afraid we'll have to stop here. Thank you
Thank
very
you so
much
much
again.